The current treatment is still radiation therapy with temozolomide, followed by temozolomide. And that's the standard of care treatment. There are many other research treatments going on, but we have not identified a good replacement for standard of care yet. So there's a lot of clinical trials with cytotoxic drugs, now small molecule targeted drugs, as well as metabolic modified drugs, and some immunotherapy approach. But these are all in phase 1, phase 2 stages. Right? Webb? Yeah.
Right, and most of these early stage, phase 1, phase 2 ones, they can be for newly diagnosed GBM. But most major trials, bigger trials, are not for newly diagnosed GBM. They're for recurrent GBM. So there's a paucity there, really, except for GBM Agile, which we'll come back to later. There are a lot of experimental therapies that are either being developed or being used, IDH being one of those, because IDH is now sort of a discriminator as used by the World Health Organization in its classification of brain tumors. It classifies GBMs into two different types, IDH mutant or IDH wild type. And the IDH mutant is the one which the IDH drug that exists is being used for. But that is not the majority, that's a minority of GBMs. So most GBMs are GBM wild type. That's the definition of them, actually. So there are some drugs like that which are being used either for a small number of GBMs or experimentally in earlier stages, early stage clinical trials.
Since we talk about the use of IDH inhibitors, and there is several really interesting leads for low-grade glioma as well as pediatric high-grade glioma that have very well-known mutation. For example, the BRAF V600 mutation occur more in a higher incidence in pediatric tumor. So MEK inhibitors is now being used for that group of tumor, and it's had some very promising results.
But that's a small fraction again, right?
It is, but it's small fraction. But that also speaks for the success that we could have if we can identify a driver gene and you have a drug that targets the driver gene, get to the tumor, inhibited the driver mutation. And that, again, that raised a lot of interest and hope in pediatric brain tumor community. Because we know a lot more of several driver mutation in pediatric brain tumor.
The most current ones are the ones that Al mentioned, which are the smaller kind of trials, right? So vaccines are one. Various sorts of immune modulations are others, which are all being addressed. I mean, immune modulation is being tested in every kind of tumor. But in GBM, these are probably relatively small and mostly in, just because of numbers, in the newly diagnosed setting. There's a lot of targeted therapies which are always coming through to test. The issue, of course, with them is that many of the targets of targeted therapies are themselves relatively rare, either between tumors, so the number of tumors that have it at all are small, or they're heterogeneous within the tumor itself. So you've still got that issue also. So they're probably, if they're successful, they're either going to be used in a very small fraction of tumors, or they're going to be used in combinations with other therapies.
So if you go into clinicaltrial.gov in the US, you probably see about 70, 80 trials,
Open trial.
trial, for brain tumor, from lower grade tumor to GBM. Most of them, probably about 80 or 90% of them, targeting recurrent tumor, not newly diagnosed tumor. And if you try to categorize those agents, I think one very focus area of research is how to modulate resistance and enhance the activity of temozolomide, or bring in the DNA damage repair agents. So that's a large group of trials in that category agents, DNA repair modifier to enhance the effect of radiation and temozolomide. Now, the other big group of agents is that immunotherapy agent. Now, because we know the checkpoint inhibitor that work in melanoma and lung cancer does not work in GBM. So there's a lot of work that's being done in developing other molecules, combination of the checkpoint inhibitor with temozolomide or with other. That's a big category of agent. The other big category agent is opening the blood-brain barrier. Combine temozolomide or combine other drug with drug that open the blood-brain barrier. So you see the trial in that common trial. The newer category that's coming up is how to modulate the metabolic environment of the tumor, targeting glycolysis and targeting the protein metabolism and modulate the oxygen environment, whether change it from hypoxic tumor to more oxygen tumor. So that's in our category. Those are the common agent or the common category of agents that we develop that you will see in the clinical trial.
This has actually been done with DIPG, for example. So DIPG is a relatively rare pediatric tumor. And Australia has played a major part in that, in both developing systems to test drugs and clinical trials and so on. But that's an international effort, which has been driven by a couple of foundations. So that part is already happening. The clinical trials are across countries. The research results are across countries. So that's really developed because this is a relatively small group of investigators who basically know each other, which is sort of the nidus of how this kind of thing would happen. For brain tumors in general, I would say that the majority of brain tumor researchers probably know most of the brain tumor researchers too. They wouldn't know them all because there's more of them, but they would know a lot of them. To have collaborative efforts, you really need a couple of things. One is you need people who are interested in whatever they're going to collaborate on. So they're not just going to collaborate on brain tumors, they're going to collaborate on iron metabolism in brain tumors, something like that. I'm making that up. So you need people who have common interests, but not overlap. They can't be the same because then it's competition. But you can have interests which are interrelated, different aspects where things can happen. An example of this is the Cancer Research UK Grand Challenge grants. So the Cancer Research UK, which is the largest multi-cancer, not just brain tumors, of course, but they set up a big grant program where the money is large, 20 million or so, for addressing some problem, whatever the problem is, which is then evaluated by a big committee and so on. And those grants are collaborative in the sense that there's usually 5 to 10 investigators, they're in different countries, and they do different things, and they run like a lab without walls. They're sort of everywhere. Those have been pretty successful. So you could imagine, for example, since you asked about CBCF, that if somebody was generous enough to give CBCF a huge amount of money, that programs like those, maybe smaller than those, but like those could be set up with investigators from Australia, investigators from the US, from Britain, wherever, coming together for a problem, a problem like metabolism in brain tumors, or immunotherapy in brain tumor, or whatever you would want. And we could set up a competition for that, which could set the parameters of the international part of that, and who did what part and who owned what part and all that sort of thing in the contract documents. That would be fairly easy to do because there are examples like that. That might be a very interesting thing to do, and I've been talking to the foundation about that, because to me it seems that this would be a great way to get young people trained, because they could go from one lab to the other of this consortium, get trained, get to know all the people in all these different places, and then come back to Australia and set up a collaborative lab of their own with all of these people that they've met along the way. So it could be a real, I'm thinking in plants now, sort of a germinating center, right? It's like be plant to plant, and it really could go there for less money if it was done in a collaborative way with other funding agencies, for example, in England or the US or wherever, together with CBCF. I think there are ways to do that which could be very productive from a variety of ways, not just the science results, but also the sort of science sociology too. So I think it's a great question. It is something to really think about, I think. What do you think?
Well, let me give you a little story also. Back in 2013, or before that, when Webb and I and Ann Barker get together and say, well, let's have a brainstorming session, because there hasn't been anything new in brain cancer research that come up to lead into a new level of research for the finding of solution for GBM. Let's say we should have a big brainstorming session. So we invited many probably in the first session that we invited about 40, 50 investigators from the US, from Europe, and I think we included Australia back in that group also. Yeah. So the Europe, US, and Australia, and a couple of people from China. We invited them to have this big consultation, brainstorming session. And one thing that come out is on that first brainstorming session before we go to the second one is that we need to have international involvement, because even GBM is the most common brain cancer, but it's still a small number. In the US, it's only about 8,000 to 9,000 per year diagnosed with GBM. In order for us to really make an impact in clinical research, we need to combine those patients from the US, from Europe, from Australia, from China. So very early on, the discussion is how do we can foster international, global collaboration in research, in clinical trial. And so we focus on that area. And the second brainstorming session, we invite the foundation. We invite foundation from UK, from US, and also Australia. CBCF was invited and involved. And so that's the kind of thinking that we have. We need to have global, even in the foundation side, if we can get the Brain Tumor Foundation, the nonprofit, that actually can come in to supplement funding from the government. And if they can combine the forces, just like we combine all the patient base. If we want to combine patient, we need to combine the nonprofit's activity, and they play a role in that. So I think these are the opportunities. There is also, actually, there was historical evidence. several foundations in the US that combine to form a so-called funders collaborative. They together fund several groups of research, just like the Cancer UK, CR UK, with a big grant that require international collaboration. It could be done. It's just really how we can break down the barrier of collaboration. I would say Webb and I are conflicted because we are the founder. We are the founder of GBM Agile. As I mentioned, we have this brainstorming session. From the brainstorming session, the conclusion is that we need to have a platform of international, global collaboration. And we also need a platform that can do clinical trial in a very rapid fashion of identify drug that work and throw away the drug that do not work. Shorten down the time for getting the answer of whether the drug work or not, and also involve fewer patients. And so that where we kind of start think about how can we create a platform trial, and also a platform trial that can also reduce the number of patients, and that bring in the Bayesian statistic, the adaptive rate trial, and that become the GBM Agile trial platform. And GBM Agile platform was designed to be able to do randomized trial compared with a standard control arm with multiple drug treatment arm, so that we can vet several drugs at the same time. And because we use adaptive statistic, then you can enter fewer patients and get the answer with fewer patients, shorten the development time. And also that means that also require less investment from the sponsor or the drug company. We're able to reduce. Usually, if you do the old way of doing 1-to-1 phase 3 trial in GBM, you'll require about 400 patients, and doing that trial will require probably about $80 to $90 million. To do the GBM Agile way of randomizations and platform trial, you reduce the number of patients to only 250 to 300 patients, about 800. So you reduce the investment requirement substantially from 80, 90 million to 30, something. So we want to achieve that. And GBM Agile achieved those goals. And we were amazed that in 5 years, since the protocol was approved by the FDA and we get started with it in 5 years, now GBM Agile is enrolling 1/3 of GBM patients that's available, that is eligible for clinical trial.
In the U.S.
In the U.S. Welcome in the U.S. As well as now Europe is open, Australia is open, thanks to CBCF. So this, again, like I mentioned earlier, if you go to clinical.gov and look for clinical trial for newly diagnosed GBM, in the untreated setting, for phase 3 trial, GBM Agile is the main one.
Yeah, I would say GBM Agile has been a smashing success so far, mainly in identifying drugs that won't work and identifying them quickly, so that people don't have to go through there doesn't have to be multiple trials of a drug that's going to fail anyway. So getting rid of those quickly, it's what the drug companies call the go, no-go decision. And getting a clear go, no-go fast is great for patients, because it means they're not going to be exposed to those things after they're gone. That's one thing. The second thing, in my view, one of the most important parts of GBM Agile is this, this layering of multiple drugs, which are called arms, into the trial with a common control arm, and which means that instead of having, let's say you have 5 arms, instead of having 5 control arms, where those patients are going to get placebo, they don't actually get placebos, they get standard of care, but they're not going to get the additional drug. That number is, if you have 5 arms, it's 1/5 the number. So most of the patients are getting the experimental drug rather than being randomized into the control arm. And that, from what we learned from the patients, was one of the major things. They didn't want to be randomized into the control arm because that's going to be what they were going to get anyway. So why should they go through all this? But the answer is you can't do a trial without a control. You have to have a control, or you don't know if anything worked. But if we can minimize the number of patients that are randomized to control arm, then more patients get a chance to go through that drug and see if it actually does good or not. And that, I think, was a major advance for GBM Agile, and certainly is probably the reason, as Al said, that there's been such an acceptance by the patient community who want to enroll in GBM Agile, to the point that we've enrolled so many patients that we're having to sort of slow it down a little bit so that we can absorb all of them into new arms that are coming on, rather than just keeping them on the same old arms when we have sufficient patients to give statistical significance already. Because we want every patient who goes on there to have the best possible chance to get something that might actually help them. That's our major concern with GBM Agile, and that was the way it was designed. What Al didn't say is all these meetings that we had at the beginning. I'm not much of a meeting guy, but we had a bunch of these meetings, and I have to say that people at the beginning of that, and we had several hundred people from all over the world, all different persuasions. We had neurosurgeons, we had oncologists, we had scientists, we had engineers, we had software developers, we had drug companies, we had all kinds of people. And I have to say that they were not unified in their view, that's for sure. It took quite a while to get everybody to realize that the importance of GBM Agile didn't have anything to do with them. It had to do with the patient. That was really the important part. And that was the way we designed it. Brain cancer is one of the most difficult. And it's one of the most difficult for a couple of reasons. One is that it's heterogeneous. So the cells within the tumor are different from each other. It's regional, that can migrate through the brain. So surgery is going to be a problem. Surgery's done, of course, but I mean, if the tumor is migrated to the side of the brain, you can't do surgery on the entire brain. That's one problem. The second problem is that it grows quickly and changes quickly. That's the multiforme part. And the third is that it's behind the blood-brain barrier. So, we've developed a blood-brain barrier as humans to keep noxious things out of our brain, right? So you don't want to have environmental contaminants able to just go through the bloodstream into your brain. And the way that that's been done evolutionarily is to develop the blood-brain barrier. But we're trying to put noxious things into the brain. Right? We call them chemotherapies, but we're trying to get noxious things across the blood-brain barrier, and to try to get that open enough to get a drug in, but not open so much that we're endangering people. There's a lot of stuff going on there. And I think, so there's a couple of answers to your question. One is, we need something which isn't limited because of this heterogeneity, right? So something which can attack regardless of what the genetic makeup is. We need something which allows the delivery, because we don't want to have to deliver over and over again into the brain. We need to be able to do it in the blood, through blood, for example. Or even orally would be better. But it has to get across the blood-brain barrier, and it has to not have toxicity elsewhere in the body. So that's another problem that needs to be solved. And the third is, we need to be able to put things in, and Al mentioned this, that are capable of stopping resistance. So we don't want to just attack a tumor, get a response, and then it's all resistant, it doesn't matter anymore, and by a few months, which would be great if we could do that, but we haven't got that yet. But there's a lot of research in each of those areas now. So the short answer to your question is, I don't know. I guess the optimistic side of me would say, oh, it's all going to come together and next week we'll have it. But I think the more realistic side of me says it's going to take a while, because each of these are problems which are huge problems. They're like going to space. Each one is like going to the moon. There's lots of parts to each of them, and each of them has to be tested, and in the end, they have to be tested in patients. So patients who are participating in GBM Agile, or it's whatever's coming after that, terrific. We're really grateful to them.